Introducing Anti-SARS-CoV-2 Rapid Test, a lateral flow immunoassay intended for the qualitative detection and differentiation of IgM and IgG antibodies to the SARS-CoV-2 virus in human plasma from anticoagulated blood or serum. This test currently does not have approval to be performed on whole blood or finger prick blood. On April 24, 2020, Hardy Diagnostics received emergency use authorization from the United States Food and Drug Administration. The Anti-SARS-CoV-2 Rapid Test is intended for use as an aid in identifying individuals with an adaptive immune response to SARS-CoV-2, indicating either recent or prior infection. This test will be useful in identifying asymptomatic or mildly symptomatic carriers of the SARS-CoV-2 virus. It will also be helpful in identifying persons who were infected by the virus previously, but may not have been properly diagnosed. This chart shows the approximate appearance of the viral antigen, IgM antibodies, and IgG antibodies. The immune system responds with IgM antibodies about 1 week after symptoms appear. IgM antibodies will disappear after about 3 weeks after the onset of symptoms. IgG antibodies appear about 1 week after IgM antibodies appear and will last much longer in the bloodstream. How long they will last and whether or not they will afford long-term protection remains to be seen. Actual patient immune response will vary between individuals. The kit includes 50 individually wrapped lateral flow cassettes as well as running buffer. No other equipment is needed other than the usual phlebotomy supplies and a pipette. The devices can be stored at room temperature and have 6 months of shelf life from the date of manufacture. This test is moderately complex and can only be performed by CLIA licensed laboratories. The procedure should be performed as follows. Collect the blood sample through venipuncture, then centrifuge the sample. Add 5 microliters of serum or plasma into each sample well using a calibrated pipette. Then add 60 microliters, 2 drops, of the buffer. Read the test results between 15 and 20 minutes. Do not read the results after 20 minutes. Please note, sample must be added to both IgG and IgM wells. Allow the test to sit undisturbed for 15 minutes at room temperature, then observe the 2 colored lines. If only the IgM test line appears, the test results indicate the presence of IgM anti-SARS-CoV-2 antibodies. This could indicate an early stage and active infection. If only the IgG test line appears, the test result indicates the presence of IgG anti-SARS-CoV-2 antibodies. This could indicate a past infection that induced active immunity. If both IgM and IgG test lines appear, the test result indicates the presence of IgM and IgG anti-SARS-CoV-2 antibodies. This could indicate a later stage active infection. Note, the intensity of the color in the test line will vary. Therefore, any shade of color in the test line region should be considered positive, even if faint. If control lines are present in both result windows and no test lines appear in either IgG or IgM test line regions, the test result is negative for both antibodies. If control lines do not appear, the test result is invalid, regardless of the appearance of the IgM or IgG test lines, and must be redone with a new cassette. Note that results from antibody testing should not be used as the sole basis to diagnose or exclude COVID-19 infection. Further antigen testing, clinical observations, patient history, and epidemiological information must also be taken into consideration. Please refer to the instructions for use included with your kit for full details. For more information about the Anti-SARS-CoV-2 Rapid Test, as well as other products related to the coronavirus pandemic, please visit our website at hardydiagnostics.com/COVID-19. Patient care is what you do. Delivering better tools to you is what we do. Saving lives is what we do together. We're here to partner with you as we work together to diagnose and prevent disease.